So DNA, there's only one version. It's just the double helix version. It looks like a double helix. It's got the same nucleotides. It's very simple. DNA is very simple. RNA is a little bit more complex because it occurs in different shapes and sizes and different functions all across the body, all across the cell. So the first one we talk about is the type of RNA that helps make a ribosome. You may remember from our organelle discussion, the ribosome is like a little tube subunit piece. The top of it is our RNA. The bottom of it is just a little piece of protein. So that's why we call it ribosomal RNA, or rRNA. The R stands for ribosome, because it's literally making up the ribosome. So that one's not incredibly, we don't talk about that one as much. I just want you to know that the ribosome is made up of rRNA. The second one is transfer RNA. So what do you think transferring, or what does transfer mean?
Like moves it to the nucleus to the cytoplasm?
Maybe not from the nucleus to cytoplasm. It transfers things. Okay, so the one that moves it from the nucleus to the cytoplasm is a message. That's a message, and that's the messenger. So the T for transfer RNA is going to be representing a transfer of something. And that something is an amino acid. So it transfers amino acids is the short way of writing that. Okay, that's very, very important to know. The T for tRNA stands for transfer. The third one is messenger, so mRNA, and that's our message. That's our little mailman that's going to go to the nucleus and say, okay, unzip, let me see the genes, let me read them, I'll make a copy, then I'll go to the ribosome to make the protein, or help make the protein. So it's transcribed from DNA, and that transcribed word is going to be referring to the first step in protein synthesis, so that's why I listed that there. But it's going to contain a code, and that code is going to help us in the end tell the tRNA what it needs to bring in terms of amino acids to make the protein. So they all work together in the end, but there are three distinct types of RNA. All right, so the messenger one is the one we're going to talk about first, and that's, it's labeled M for mRNA. All right, other differences between what we talked about last week and this week. DNA, deoxyribose. RNA, ribose. Sugar is one specific thing that's different. DNA, double-stranded or double helix. RNA, single-stranded, right? So the DNA is double, the sugar is deoxyribose. And then we have one base pair that is different between the two. So instead of having A to T, C to G, that's for DNA, what would the A be pairing with according to this statement? U for uracil, and then the C and the G are still cool. They're still the same. So those are other three differences between RNA and DNA. And before I forget, you have a RNA DNA comparison homework, online homework 1. Pretty simple. It's online. You'll see it on your homework for tonight. Since you guys got the day off tomorrow and all. Yay. Okay. So those are the differences. I have another chart that's got a comparative mRNA types and the function, but I want you to add the location. That's the only thing that's not on here yet. You can probably infer the location because you know what they do a little bit, but I want to make sure. All right, so here's what mRNA looks like. If you like to draw it. You notice that it's just a single strand. It's got little prongs on it, which represent the nucleotides that are the nitrogen bases. It only exists in one place, or two places really. It can go to the nucleus, and it can go to the cytoplasm where the ribosomes are. So if I say, what type of RNA is able to enter the nucleus, you would have to say mRNA. None of the other ones can enter the nucleus. That's the only one. So that's why I want you to write down the location so you don't forget that. And that's the one that's carrying the information from DNA to the ribosome. The second one's got this weird shape, but if you turn your head to the side a little bit, what letter does it kind of look like? A T for tRNA. It's kind of a T, yeah? Yeah? Okay. It looks like a fork, but that's actually what it looks like. It's a big, huge, long strand crumpled up into that shape. And so that's how, if I see that, I know that's tRNA, because it looks like a T. And that one cannot be in the nucleus at all. It can go and hang out with the ribosome, but commonly you're just going to have them floating around in the cytoplasm. Can I answer any questions? Manisha, anything? I said that you can find it in the nucleus and the cytoplasm. So I said, if I asked you what type of RNA could you find in the nucleus, the only answer would be mRNA, because that's the only one that has the ability to penetrate and go into the nucleus, or go through the nuclear pores. All right, and tRNA is working with transferring stuff. We'll get to that shortly. rRNA, guess where it's found? Where? Oh, it is found in the cytoplasm, but at the ribosome, because it is the ribosome, okay? It is the ribosome. So it's going to be found at the ribosome. And that's what this shape kind of looks like. The top part are RNA and the bottom piece is a little piece of protein. Thumbs up when you're good here. I'm going to be passing out stuff. Don't be too distracted. I'm going to try to pass out your test while I walk and talk here. Okay, so let's talk about the next part of the whole protein synthesis segment. The first step of protein synthesis is called transcription. You can abbreviate these steps, but I give a lot of detail so that there's no questions about what you need to know about this. So this right here is step 1 of protein synthesis. Take a minute and write down the individual pieces of this overall first phase. And I'll give you some information.
So that's all step 1, but there's other steps.
Yeah, these are the pieces of step 1. Or these are the steps of the first process. Yeah. Try not to get too distracted by your test. Oh, that's not please. I get used to your guys' news. Alright, so if I were reading this, first thing I would see is that the teacher has highlighted the C up here. It's a different color. It's going to be important to help you understand and remember the two different processes. So a transcription has a C in it. Just make a note of that. Alright, the very, very first thing that has to happen is DNA has to allow for the code to be read by something, and that's going to create the message. So the very first thing is to make the message, which is mRNA. So this right here is the overall goal of this process. The very, very first thing that has to happen is DNA has to let something in. So it has to open up and unzip. So that's pretty easy. Then RNA polymerase, the enzyme RNA polymerase, well, we had DNA polymerase, and that was to make another copy of DNA. Well, RNA polymerase is the same type of enzyme. It's going to make many RNA nucleotides strung together, but it's just RNA because it's dealing with RNA nucleotides. So the RNA polymerase is going to allow for free RNA nucleotides to come and bond to one strand of the DNA. Because, I mean, you got 2 copies of DNA, they're complementary to one another. It's kind of dumb and a waste of resources to copy both of them, because they're going to be coding for the same thing. So don't do it more than you need to, because you're wasting your valuable cellular resources if you have to do it twice. So it only goes on one strand of the DNA. And then I said honors because I asked for you guys to know the enzymes. Last and third step is the RNA, once the mRNA strand has been made, it has to be able to leave the nucleus. It's still attached to the DNA at some point, so it has to break off and then send itself out of the nucleus. So all that together means overall goal, make mRNA strand from DNA strand. But after this, it's going to be going to the cytoplasm for part 2. Thumbs up if you're good here. couple of us. Yes, ma'am.
You know, at the top of your row, it was like step 1 of protein synthesis.
Mm-hmm.
Is that just a bullet point, or is that
No, this whole process is the first part of protein synthesis. Part 1 of protein synthesis.
So
It's called transcription.
Okay.
Yes,
Why does it say DNA from one gene is transferred?
that's what I'm going to talk about. So, while some people are finishing that. So, diabetics need to make insulin, right? Everybody's going to make insulin. Well, diabetics just have a deficiency in the ability to make insulin. Insulin is a protein. So, what is going to be coding for that protein? A gene, right? So, if one of your body's like, oh, just ate a big healthy Thanksgiving meal, and you guys start getting that glucose into your cells, your body cranks up the insulin production. So what has to happen is it goes and finds the gene, just the small part of the DNA that calls for or codes for insulin. So what they do is they go and find it. They send RNA polymerase and say, all right, this little this small section, I need you to make some of this. So the RNA polymerase goes there and it reads just that small section of DNA, and it's going to allow for mRNA to code for the insulin. That little mRNA strand pops out of the nucleus, goes to the ribosome, and it goes to the ribosome and actually literally makes the protein insulin. And it does that, 100,000 times, 200,000 times, a million times. Doesn't matter. It's going to make a ton of it based on the amount of insulin you need to make. Your body codes or tells it to do so. So most of the time, what we're talking about is making one protein at a time, and that's calling for one gene at a time. So it doesn't make a ton of them at once. It codes for the things your body needs. Okay. And then we know the mRNA has to go to the nucleus because the DNA does not move. It's lazy. All right, so if this is my DNA strand, what is my mRNA strand that's being made? Go for it. A? No. U for uracil. Remember, mRNA does not have any T's in it. Anytime you want to say T, just hold back and say U. Okay, keep going.
C, G, A, U, C, G, A, U, C, C, G.
Okay, so this right here is the mRNA strand that was made. It breaks off, and it goes where? To the cytoplasm. Okay, so that's what we're going to talk about in a second. All right, I'm going to skip that for now. And take that. Okay, so before we go on, though, we have to be able to talk about the parts of the mRNA strand that are important, and the parts that are important are called codons, because they code for something. What the mRNA's job is to do is say, here's the DNA, code, tRNA, bring me the amino acids. So they have to break them into the sections so they have a specific nucleotide sequence for the amino acids. So the codon is a sequence of 3 RNAs on the mRNA. So I'll let you copy that down in just a second. I'm going to go back here, and we're going to look, and you tell me the first codon that you see on the mRNA strand. Okay? So what is the first codon you see? AUC. Codon, that's the first one. Next one. Here. And then we don't have a full one on the last bit there. So how many codons do we have here? 4. Okay, these 4 codons are going to tell tRNA, bring me glycine, which is a type of amino acid. Bring me serine. Bring me tryptophan. Bring me phenylalanine. Bring me whatever the amino acid is calling for. Okay, so these are coding for that. But the tRNA doesn't know where to go, really. It just knows it's supposed to bring it. What it has to do is the tRNA molecule, which looks like a T, right? Here's my little tRNA. Yeah. Okay. The tRNA has to know where it's supposed to go, so it has to go and fit the pieces together. The only way it knows where to go is it base pairs with the codon. So what would base pair with the A here? Still dealing with RNA. U, A, because U is like a T, so A pairs with that, and G. Okay, so the codon base pairs with this called the anti-codon. Okay, so that's all on the next slide I'm going to show you. I just want to show you a picture first. What was the job of the transfer RNA? What was it carrying around with it? Look back at your 5 seconds ago notes. It carries amino acids. Thank you, whoever said that. Okay, so this is going to carry amino acids. So it's going to have on the bottom of it some amino acid that it's carrying around. So it has to find the codon, and the anticodon base pairs with it. It brings the amino acid to the mRNA. All right, and that's what I had right here. All right, so the codon is the first 3, is a sequence of 3 RNA nucleotides on mRNA. sounds like I'm speaking on like random code because I just say letters, but they mean things to you. That's why this unit's very vocab rich. So it has to be the one, the codon is on mRNA. And then the anticodon and T codon is on the tRNA. That's how I've always remembered it since I was ninth grade or something like this. The anticodon code is on the tRNA. And they complete each other. The mRNA codon is completed by the tRNA anticodon. And that helps us get that final step, bringing the amino acids to the ribosome for that final step of protein synthesis where we're going to write some information about that.
Is it codons if there's like a leftover one, or is it just like you and there?
Usually what happens is that example was just there, but usually what happens is the mRNA makes exactly the number of codons it needs to make the protein. It doesn't have leftovers.
Would there be like, would something go wrong?
If there was an extra, it was probably due to a frame shift mutation. You're going to see more about tomorrow the next day.
Okay.
It's because something else went wrong before it got to this point.
Okay.
Yeah, which would be bad. Well, not all the time. Sometimes mutations are good. I say that, but sometimes they're neutral, sometimes they're good, sometimes they're bad. All right, and then I have just a picture of the same thing. We saw these codons already. So what would base pair? What anticodon would come into base pair with AUG? What? UAC. No T's. It's okay. You just learned that like 10 minutes ago maybe, so I let you go. Huh?
I don't understand that part.
You base pair, so in RNA's period, it doesn't matter what I'm dealing with, mRNA, tRNA, or RNA, the A and U pair together and the C and the G pair together.
Yeah, I get that part, but like, why did you get that way?
Oh, what'd you say?
No, I get that part, but like, why is it in that order? Like, AUG, how is that UAC?
Because what I was asking for was, my question was, if this is the codon, what is the anticodon? That's what I asked. And so the anticodon is complementary to it, it base pairs with it.
Oh.
So that's what I was talking about. The anticodon is from the tRNA, so this would be something on the tRNA, and the tRNA would have a little amino acid it was carrying around with it too.
Oh.
So I think I have a picture on the next slide I think will help. All right, so here in the background is the ribosome. The ribosome has this little thread through it, which is your mRNA. Well, that's the messenger that brought the DNA code to the ribosome. So you can see that it did its job. It's there. The very, very first codon from the mRNA was AUG. This is called the start codon. How have I remembered that for like, I don't know, 15 years? I remember that as the start codon because we start school in August. Okay, the start codon says, all right, ribosome, start making a protein. The ribosome won't do anything until it sees AUG. Okay, so that's something you need to write down. The start codon is AUG. August. That one's easy because it's August. It's the first day or the first month of school. All right, so the very, very first, and you can see on the bottom too, the very, very first start codon is also going to call for the first amino acid. But the first thing that has to happen, it's already been done, so I'm going to actually skip that because you can't see anything. So let's say it's already started the process. Inside the ribosome right now is the codon GAA. The anticodon that's going to come and bond with that is attached to this T-looking thing. It's kind of a T. tRNA. CUU is going to base pair. That CUU is called the anticodon. This anticodon is on the tRNA, and along with that, tRNA is carrying an amino acid. That amino acid is going to get dropped off and it's going to attach to the previous amino acid that had already been dropped off by these previous codons. I'm sorry, by these previous tRNAs that were here. You see that one right there's leaving now because it's lost its amino acid because it dropped it off right here. So this is going to continue until the whole amino acid chain is built. Yes?
So does the mRNA really just tell the tRNA where and when to drop the?
Mhm. The correct amino acid, yeah. Because the gene, the DNA is coding for a specific sequence of amino acids, and since the DNA can't do it, the mRNA has to do it. Has to tell them how to go.
And the tRNA already would have like the CUU, and that would just pair up with mRNA.
Mhm.
Okay.
And in that process, drop off the amino acid that the mRNA is saying bring to me. And so this is another version of the tRNA. It's got this weird looking shape. But you can see on one end is the amino acid, and so I always call it the head, and then the feet are the anticodon. All the time, yeah.
Why have the T's changed to U's?
Because it's based off of evolution, really. It's a long story. But RNA was the very, very first form of the genetic material of life. And to allow for more complex life to form, well, not to really allow for it, it happened to allow for it. There was a mutation in the uracil way it was shaped. So the uracil could no longer fit with the adenine. And so we now call it, the mutated version of it, thymine. And that's most commonly found in DNA, not RNA. So life came from RNA, which is a form of genetic material, but it's a more simplistic version because it's only single-stranded and it doesn't form as complex of proteins. And so mutation caused it to turn to thymine, and then we had the synthesis of DNA and so forth. It's a way past, like way long time ago. Yeah.
Does that mean thymine a mutation?
Thymine, yeah, it's a positive one. Thymine was a mistake in one of the codes, and it transcribed differently. Okay. Oh, and so here's my little cheesy way of doing it. Oh, this is mine. This was really ugly, but it's there. I did that with a mouse, not my pen. Okay, anticodon tRNA. Okay, that's what I was doing stuff at home. All right, so now you do not have to memorize this. This is something you just need to know how to use. It is a tool. Like the periodic table is a tool. This is a tool for geneticists and molecular biologists. So we know what codons are, and we know what anticodons are. If I just want to look at the mRNA strand and say, what amino acids does this code for? I can do that. So you know what was the start codon called? AUG. Okay. All you do is you use a chart like this. This is called the genetic code chart. It's in that book. It's on page 292 if you care at all. I'll explain more about what it is. I want to show you how to use it first. So AUG, there are 3 letters. The first letter you have to look for on the left. The second letter you have to look for on the top. The third letter you have to look for on the side. So A is down here. So you've kind of isolated and said, okay, I know my first letter is here, so I can look at and see any amino acid in this row. But my second letter starts to narrow down what amino acid I'm talking about. So the second letter was what? U. And you can see that U is the first row, or sorry, the first column, and so forth, okay? So now I've narrowed it down to this row, but this column. So I've narrowed it down to this quadrant. Or I guess I'll say quadrant, it's not really a quadrant. Narrowed it down to here. And then sometimes there are little codes are listed here, like right next to the amino acid. But if it's not listed, you have to look over here. UCAG. Our last letter was a G, so you take the G and you move over to where you were. So the first one is the start codon. So it says start. It also says methionine, and that's what the very first amino acid is for all of these. For anything that has to start. All right. So if I wanted you to tell me what amino acid something called for, let's do this. What's CCC? Proline, good. So you went C, this row, C, this column, so you're in this region. And any of these, U, C, A, or G would have called for the same amino acid, right? Right, so I can have a mutation where instead of going CCC, I went CCU or CCA or CCG. I could have a mutation, and would it make a difference in the end? Nope, I'd make the same exact amino acid and then the same exact protein. So this genetic code allows for mutations to happen because proofreading, like, I don't know, 200,000 base pairs, is not going to always be exactly right. Just like when you read over a paper, it's not always going to be exactly right. You're going to miss a period or a comma or misspell something, spell check thinks it's still a word or something. So it happens, but it's okay because the genetic code is allowed for mutations to occur because it's redundant. The same exact amino acid can be made from different types of codons. All right, now I want you to write down the top part here because this is important for evolution and classification we're going into next year. Give you guys a stretch break in a second here. So you've probably heard that all life came from a common ancestor, maybe, or evolution of species based off of Darwin's observations. But this code, this chart that determines what proteins are made or what amino acids are made, is the most commonly supporting evidence for evolution from a common ancestor or single origin species. So DNA, we know, has 4 letters, A, T, C, G. Our DNA has that. What about a plant's DNA? Is it different or the same? It's the same. A bacteria's DNA is the same, ATCG. Yeast, ATCG. Every living thing on the planet has exactly the same 4 letters in its DNA. Now, those 4 letters can be put into lots and lots and lots of different arrangements to code for different types of genes. That's no problem. Thinking about the length of our DNA, that's not really our limiting factor. It's like, how long can you make the DNA? There's so many different arrangements of A's and T's and C's and D's you can make. That's why life is so different. But this ATCG scheme or sequence says, well, we all have the same inherent molecular structures, then we probably came from a common ancestor. And then furthermore, the amino acids that are made, there's only 20 different amino acids. Another note that you might want to write down. There are 20 different amino acids in your body, in a plant's body, in bacteria, in a tree, whatever. So those same 20 amino acids show up in every form of life, further suggesting that there was life that was formed from one common ancestor. And that's why we talk about this one a lot too. All right, give me a minute, stretch break, because we need to go over translation. Take about 5-10 minutes, and then let you guys practice. Get up and talk to somebody about genetics. I'll call you out for your test. Come see me for your test. Taya, Michael, Sarah, Nisha, Jackson,
Mallory selfie with
What were you asking me?
What'd you get on your test? What'd you get on your test? What about you, Joey? What did you get? What'd you get, Joey? You briefed that, dude. No,
I think that's fun. They were looking at us and were like,
All right, back to your seats. 5, 4, 3, back to your seats. Sorry.
oh, whoa.
2, 1. Stop codons. I'll give you a second. Misa, student want to take biology. Put your test down right now. You can finish looking at them later. go ahead and write down the stop codons. Those are more that you need to know. August is easy, these are not easy. You need to write them down so you know. So if you know that the start codons are going to say, okay, transcription, translation, let's go ahead and start, then these are going to tell it to stop. Because if not, it would go on indefinitely until the mRNA ran out. And sometimes that's okay, but sometimes it needs the ribosome needs to know when to stop calling the tRNAs over and saying, please go ahead and make this protein for me. So that's just going to tell us where to stop. The next thing you're going to write down is something about translation. And it's long, and I'm going to write down the important things on the board. We've already talked about it a lot.
The start and stop codons are both on mRNA, right?
Yes. Good point there. So these are on the mRNA molecule. But if I go back to the codon chart, pick one of the tRNAs, pick one of the stop codons you're going to look at. Which one? UAA. Okay, UAA. UAA. It just says stop. It doesn't code for anything. So those are pretty easy because like you can actually look on the codon chart and see what they are. If you have the codon chart in front of you, that is. So you could backtrack and say, oh, well, let me see what this one is. Well, this one is UGA or UAG. If you can find it on the codon chart, you can do it. You can use it. All right, so translation is going to be a little bit long. I'm going to first introduce like what the goal of it is, and then we'll go through the parts of the process. So translation, the first thing I wrote was an L, capital or highlighted L there. I did that earlier with transcription, because once we say transcription, translation, you're starting to get your words jumbled together. You're like, oh, I forgot which one's first, which one's second. I remember transcription is first, and translation is second because it's alpha order. So that's the only reason I did that, to give you a little tip on how to remember which is the first process and which is the second process. So transcription and translation. The main goal of translation is to take the mRNA code and to bring the amino acids together for a protein. That is the main goal. It takes a little bit to do it, but we've talked about it a lot, so I think this will make sense. So today's note heavy. And then you know where it occurs, so we've talked about it a little bit, but this protein synthesis, the finalization of it has to happen at the ribosome, which is in the cytoplasm, usually. And then when does it happen? After transcription. And then I just reminded you that the amino acid chain, a huge long chain of them, is called a polypeptide. And that's just a term that we've talked about before, but I wanted to remind you because you already knew what it was. And it might say, like, what process finalizes the production of a polypeptide. And that just means, what's the second step of protein synthesis, but the word polypeptide sometimes throws people off. Okay, do not be scared. Let me tell you what's important here. You can see that the process of translation is multi-step. Step 1 through 5, okay? But let's read it, and let's write down the important things. Because I think you'll find that you already know all of this. Step 1. mRNA attaches to the ribosome. Translation starts with start codon. Did you already know that?
Nope.
Yeah, pretty much from the picture. So step 1. mRNA at ribosome start AUG. Start codon. Write down, of course, everything you want, but I'm giving you a simpler version. Step 2. tRNA brings the amino acid to the mRNA anticodon bonds to codon. Okay, tRNA anticodon bonds brings amino acid. And you, if I say the tRNA anticodon bonds, you're like, well, what is it bonding to? You know it bonds to the codon. And so that's where your previous knowledge comes in. Step 3. Continue. Continues until what? That same process. tRNA comes in, takes its anticodon, bonds, delivers amino acid. tRNA comes in, bonds, brings us amino acid. Blah, 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 blah. It's going to happen until it sees a what? Stop codon, right? So it continues until, I'm just combining steps 4-ish, 3 and 4.5, I guess. Continues until stop codon. This last part is the only part we've not really discussed at in depth yet. So step 4.5. After the stop codon is reached, that symbols or signals to the ribosome, we're done here. Let's go ahead and deattach everything. Just let everything go to its normal place. When that happens, though, what was being made at the ribosome? Amino acids were being strung together. So it was like one amino acid got dropped off, and then it attached to the next one that was being dropped off, and then it attached to the next one that was being dropped off. So it's a bunch of amino acids that are being linked to each other like little beads. And when that process is done, it's reached stop codon, it breaks off from where it was at the ribosome, and it folds. It folds into a very, very specific shape. And you know that shape matters in proteins and enzymes because sometimes, you remember, we had the substrate and the enzyme, if they didn't fit together, they wouldn't react or they wouldn't catalyze or something like that. There's the shape of this is very, very, very important. So that's the only part that I would definitely want you to write down. Amino acid chain will break off and fold to form that protein structure. Because that shape is really, the shape is important because controls the function of the protein. And that function of the protein is the real reason we're making this protein. It's not for kicks and giggles, like, ooh, I need some more hair, let's grow some more protein, or my fingernails are short, let's grow some fingernails protein. Well, most of the time the proteins that would come out here, like structural ones that are building muscle, or making enzymes, or controlling the pigmentation in your eyes so you can see, stuff like that. All right, after the fact, when everything's done, the ribosome and the mRNA break down. They can be reused. They're not going to like go to the lysosome and get trashed. The tRNA is going to return to the cytoplasm and go find another amino acid. And it's going to hold on to it until protein synthesis starts back up again. So the last step is kind of like just deassemble everything. Thumbs up when you get here. I'll give you guys another second. Yes, ma'am.
Step 4, which is underlined.
Excuse me?
Step 4, the one which you underlined.
I did step 4 here and there. Yes, ma'am. Underline.
Thank you for showing your patience.
Alright, I will come back here if I need to. But I see a lot of cases are like, we're ready. So I usually go with majority rules. Alright, so here's what this process looks like on a cartoon version. Here's your what strand? mRNA. What's the big blue thing in the back? Ribosome. What are the squiggly things?
tRNA,
tRNAs. What is that?
amino acid.
Amino acid. And then what is bonding to the codon? Anticodon. And that's all in the cytoplasm too. Okay, and then here's it again. DNA's double helix makes that single strand of mRNA. It goes to the ribosome. It's going to be threading itself through. tRNA bonds, and then the 1, 2, 3, 4, 5, 6, 7 amino acids have already been dropped off to make that polypeptide protein. And then I'm not going to do that. Here's it again. But I was going to show you the video clip. That's what I was actually doing. I need to go back and show you one. I didn't show you a video already, did I? Okay. Okay, let's do this one. Actually, I already had this pulled up. That's why I had it pulled up. Okay, will you cut the lights for me? One of you guys? Thanks. All right, and this is going to talk through like the whole process. This is the extreme molecular value. Or,
How a simple code is turned
Turn on for me.
into flesh and blood. This is what Francis Crick called the central dogma of modern biology.
that's DNA
How DNA
proteins on the
makes
end of it.
protein.
getting ready to make
It starts
DNA.
with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left.
There are like 5 enzymes that help do this. I'm only telling you one. There's something called Okazaki fragments, which you'll see in your video. No, you saw that in your video on DNA. Yes, do you remember this? Okazaki fragments. And ligase and a couple of other things, but I'm not going to ask you to memorize those, so that's why I'm not telling you everything that's going on. Yes, ma'am.
Why was it like twitching?
Because they have kinetic energy, which is energy in motion, and they continually move until they proceed with the process.
Everything's ready to roll. 3, 2, 1.
So it flies through the double helix. And that's
The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA
What's your
to
name
form a
on it?
miniature factory called a ribosome.
Sorry, it's from
It translates the genetic information in the RNA into a string of amino acids that will become
What's
a
flying to
protein.
tRNAs, yes.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, You are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
Okay, cool. Let's really quickly do our own, and then I will leave you alone. I'll let you guys do an activity and look at your test too. Okay, so on the board I have a DNA input screen. You guys can write this down, or you can just visually do it with me. But I have to scroll back to the codon chart. Whoa, I just skipped something really fast. Okay, I'm gonna leave this up here. Here's my DNA template strand. What is my mRNA strand? U. So A and T pair together, right? So that T will still pair with an A. That is still true, okay? Right. So essentially what you're doing is anytime you want to write, like you thought O-A-T, and you want to write a T, just replace it in your mind saying U. So don't get confused by that. All right, so these individuals are called what now? Codons. Those codons, you take the codon chart, you look for the next thing, which I'm asking for is the amino acid. So everyone should know the first one is going to be the start codon, right? That's why I intentionally did AUG. What amino acid did that call for on the codon chart? Methionine. A little bit meth there. Methionine. Okay. GGU. Can you see it? I can't see it, so I have to look. Glycine, I believe you guys and stuff. Okay, glycine. Gly. Next one's CAU. Is it histidine? I do believe you, but I gotta look. C. I was looking at CAU. Is it histidine? So CAU. Okay, yeah, histidine. Last one. UUA. Leucine. Okay, and I could go on and on and on, but what I would have to have at the very end is a stop codon. So, like UAG or something like that, or UAA. Okay, so then this right here, I could ask you, and the only reason I did this was I want you to know directly, take your codon, find your amino acid. There is an anticodon, present here, but I do it at the very end so you don't accidentally look on there for anticodons, because that's not an anticodon chart. That's a codon chart, okay? Because it would be opposite if you did it that way. So what would be the anticodons for these? U, thank you. Good job, guys. Yay, you got yourselves in the bag. You just made a protein. I'm going to say that's insulin. Okay, here's what I'd like to do for the last 30-ish minutes. There's an activity where you are going to transcribe and translate your own protein. You have to do it in a partner though, but I had to make a lot of copies, so I do it in partners. This is a huge strand of DNA. There should be a partner strand here. I've already unzipped it for you, okay? So I've done the very first part. There are nucleotides here that are all DNA nucleotides. You're going to take and make an mRNA strand based off of this. I already have everything cut out for you. You're going to make the mRNA transcript. Then you're going to go to the ribosome, and you're going to make the amino acid translation. And then we're going to talk about the protein A, okay? You may not get done with this whole thing, and that is fine. We'll have some time tomorrow. Also, I will have the test papers up here. If you would like to look at your test, do so. If you did well, you did well. Look at like for 5 minutes of the questions that you need to. I will have them up here though, just so I want you to start on that, and then you can come back and do the test if you need to. So look at the person you want to work with. I know this is how this works. And let me tell you what I need you to get from back here. So, this is your DNA strip. It is labeled, look up here, please. Thank you. It's labeled 1, 2, 3, 4. You have to cut it out and tape it together to make one big long strand. And then I put little notes here, like you need one of these purple strands, which are Aerosol, plus one little single one, and you can get that later. You can start back out grabbing one of each color or two of each color. We should have enough for that, okay? And that's going to be you making your mRNA strand based off of that. Questions? No? Grab scissors and glue as you go. Start here and go this way. Find a place to settle down. We just need one of each color. For now, it's good. Okay, you are going to do this activity, but I need to show you. Oh, I gotta stop videoing.